Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Freddie Lawton from Business Review, and I will be your host. It is our pleasure to have Batavia Biosciences with us today, who will be discussing enhanced protein expression in biopharmaceutical development. Today's guest speaker is Femke Hoeksema, who is a senior scientist and team leader at Batavia Biosciences. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you receive via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. We will allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. If you click on the survey widget, there are a few questions in there for you to answer in order to provide feedback. You can do this during or immediately after the webinar. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now please allow me to welcome Femke.
Thank you for your introduction, Freddie. I will be talking today about the STEP technology, which is an expression platform that delivers fastly high protein expression. So, in this talk today, I will be starting by briefly giving a company introduction about Batavia Biosciences. I will then proceed into introducing the STEP technology and also explain the STEP processes and how the technology could be utilized as alternative to transient productions. I then will present a case study, the expression of erythropoietin with STEP. And of course, I will end with the conclusions, and then indeed there will be time for a questions and answer section. So to start with briefly introducing Batavia Biosciences, our company started in 2010, and it was launched by two scientists. They had been working at Crucell on antibodies and adenoviral vectors for vaccine manufacturing. And actually in their work they had encountered in their experience as scientists that there was a lack in a flexible outsourcing partner that could handle everything between a good idea in proteins or vaccines via process development and product characterization to GMP production. So their main driver was actually to fill that gap with Batavia Biosciences. And now in 2016 we work with 75 people at 2 R&D sites. Together we have some 40,000 square feet on 2 strategic locations. So one is in the Netherlands, where I am also based in Leiden, and the other location is in the US, in the Boston area. So in our company we always work with dedicated suites for protein versus vaccine and mammalian versus bacteriological. And we envision ourselves as a service company that brings experience and helps the customers in their process, and we therefore have highly experienced and educated staff, with 42% of our staff being PhD graduated. In the Netherlands we are also performing GMP manufacturing. We have a GMP license since 2012, and we are experts in the fields of viral vaccines, bacterial vaccines, and complex recombinant proteins. So at Batavia Biosciences, we add value for the client rather than commodity research and manufacturing only. And we therefore also put effort in actively developing technologies in support of the biopharmaceutical industry in bringing new or improved medicines to the market. And we offer our expertise in development of high-quality research materials for preclinical studies, scalable and robust processes to allow IND filing, and GMP materials for entry into the clinic. 3 of our current technologies in support of those activities include the STEP technology for enhanced protein production, the subject of today, the SCOUT technology, which is a high throughput technology for USP and DSP process development, and we have CIDES, which is our viral vector technology. But first now, of course, let's proceed with the STEP technology, which is the subject of this talk. And to first start with our rationale to develop an expression technology. So why is there a need for high-producing cell lines? As all of you know, advances in cell culture technologies in the past few decades like process and media development, but also improvement in expression technologies have resulted in significant increases of yields of proteins or antibodies produced by mammalian cells. There's still a need for improvement, however, since yields in mammalian cell lines tend to be low. In addition, the cell line generation process is still, in general, a long and labor-intensive trajectory, so still an area for improvement. And in general, you could say that increasing yield will have most profoundly an effect on the manufacturing scale. So increasing cell line expression is an important step to reduce process development and manufacturing costs by reducing the scale of the final manufacturing process and also the associated savings in resources, materials, facility costs, and scale-up time. So this effect of high expression resulting in reduced manufacturing scale with these associated savings in time and cost has an effect in the diverse biopharmaceutical fields. For biosimilars, it's still an important topic because of the constant pressure on healthcare systems to have significant and sustained cost reductions. And in the field of rare diseases, in which the market interest and the budgets are much smaller, an efficient cell line generation process and increased expression levels can have the effect of actually enabling preclinical studies and clinical manufacture. And also in the fields of novel products, there are, of course, still the manufacturing scale considerations, as just discussed, but also a reduction in time to select a good expressing cell line will also reduce time, for instance, for material generation, so allowing product characterization and potency and candidate selection studies. But importantly, also, if a reduction from time to market can be established, the profit in the patent exclusivity period is increased. So we had the desire to develop an expression technology which should provide all those benefits as just discussed, and it thus should enable rapid generation of high-producing, scalable, and stable CHO cell lines. A high quality of product is needed, and a resource-efficient process. The cells should be pharmaceutical grade, so animal component-free processes, and fully supporting any future entry into GMP. And keeping all those demands in mind, we developed the STEP technology. And the picture here illustrates what the STEP vectors look like. The technology is actually based on three key features. The vectors contain functionally impaired selection markers and DNA spacers that impair translation. And this creates a very stringent selection system. With a stringent selection system, you introduce a threshold level. So because of the hampering of the senescent selection marker and the hampering of translation, impairment is introduced at the protein level. And this means that only cells with very high mRNA expression levels are able to survive. A second key feature is that the vectors contain two types of novel expression enhancing DNA elements. They flank the expression cassette, and by enhancing expression, they actually enable the use of the very stringent selection, since in the absence of those elements, cells are not able to survive. A third important feature in the vectors is the IRES sequence, the internal ribosomal entry site. This enables the translation to start within the mRNA. So because of this feature, expression of the gene of interest and the selection marker are coupled transcribed into a bicistronic messenger. And I already told that stringent selection means that cells can only survive if they have sufficiently high mRNA expression levels. And since translation of the gene of interest is optimal, the end result is that there are only few surviving cells, which all show high expression levels of the gene of interest. So an important consideration for applying a stringent selection system is that you don't want to be too rigid in your stringency, since this can have the effect of no cell survival at all. And in the STEP technology, we have different senescent mutants, which are affected in their functionality to different extents. We also have the spacer elements that have hampering of translation of the CSIN marker at different extents. So we are able to adapt the level of stringency, and currently, practically, this means that we have 3-step plasmids in operation, and typically high stringency favors more easy to express proteins, whereas low stringency favors difficult to express proteins. And the effect is actually that for each type of protein, maximum expression levels can be efficiently obtained. So in the graph on this slide, you see the effects of these 3 stringency factors. And to illustrate the effects, we used a green fluorescence marker. Each dot in this graph represents the green fluorescence level measured in one cell line. On top, in orange, the numbers of the obtained clones are shown, and as you can see, the higher the selection stringency, the less cells can survive, but expression levels are increased in these cells. So standardly, in cell line generation, expression levels will follow a normal distribution. And if you then only want to check a few cell clones, because that's an efficient process, you will actually only identify low expression levels. And with the stringent step system, low expressors don't survive the selection pressure. So there's actually only a need to check a few cell clones and already identify good expressors. So this slide is actually showing some of our track record at Batavia Biosciences. As I explained, the step technology is novel, and also Batavia Biosciences is a relatively young and small company, but we have already been able to use the technology to express several proteins from several fields of interest. We generated step CHO cells that expressed 2 types of blood coagulation proteins, 2 types of bone morphological proteins, a novel pro-angiogenic protein, and 2 types of HIF envelope proteins. In the field of biosimilars, we produced blood coagulation factor 8, 3 types of enzymes for enzyme replacement therapy, and erythropoietin, EPO, of which I will also show some case study in this presentation. We also worked with 2 types of monoclonal antibodies, and in any case, we observed that we obtained higher yield with a higher speed at a lower cost than for typical cell line generation processes due to the key features of this step technology. So I would next like to explain something about the step cell line generation process, how it works, what the workflow is that we apply, and some of the timelines that we can generate. So this slide actually illustrates differences between typical cell line generation processes and the efficient step cell line generation process. So on the left, a general process is illustrated. An expression factor is transfected in host cells, after which selection pressure is applied. And the cells in which the DNA is integrated and expressed, or at least the selection marker, will then be selected. And since expression levels of the proteins of interest normally follow a normal distribution, and because of differences of orders of magnitude, of 2 orders of magnitude are typically existing between highest and lowest expressing cells, enormous numbers of cells need to be screened to identify a good expressor. This is a very labor-intensive and time-consuming process, and typically at multiple culture formats throughout the process, a reduction in number of cell lines is made based on screening results. And the process can be performed under static conditions, after which adaptation to suspension conditions is needed. And multiple rounds of gene amplification are typically used, and subsequent screening are also involved to obtain the lead candidate cell lines. On the right, you see a typical step cell line generation process exemplified. The step factors are transfected into CHO host cells, after which selection pressure is applied. And expression levels are enhanced by the expression enhancing DNA elements, and only the high expressors survive because of the stringent selection system. So since we only need to generate a few cell pools to find a very good expressor, only a few cells need to be screened. The whole process that we apply is that the cells are kept throughout this process in suspension, and the surviving cell pools are directly screened in batch productions in shake flasks under shaking conditions, which is also already directly a good predictor for the cell performance in bioreactors. So the advantages of the STEP technology is the rapid and efficient process to generate high-yielding cells. We rapidly generate pools that are already of such high quality that they are directly of great value for material generation, for instance, for candidate selection, potency studies. And as the pools are of such high quality, we typically also only generate a small number of clones. So also that screen is resource efficient. The generated glycoproteins are of high quality, and the cells show stable and sustained expression, and cells are scalable to larger production volumes. So in the EPO showcase, I will also exemplify some of these statements that I'm now making. This slide is actually reflecting timelines of our standard STEP workflow. And first we start with stable pool generation. We generate only minor numbers of pools in the efficient selection process. So from DNA, it only takes up to 5 weeks to generate and select a top producing pool that has been subjected to a batch production. This top pool can then be subjected to limiting dilution clone selection, and a top clone is selected within a further 6 weeks. The complete process supports potential future GMP using chemically defined animal component-free medium and fully traceable materials. The speed in our STEP workflow is primarily introduced by the efficient stringent selection process, and therefore there is only a need for a few cell pools to be screened to select a high producer. There's no amplification involved, and we use suspension-adapted host cells, and the complete cell line generation process is performed under suspension conditions, so no need for suspension adaptation afterwards. And because of the speed in our workflow, the STEP technology is also very well suited for rapid material generation, which we can start already after approximately 3 weeks. And this results in a competitive timeline for material generation, because after such a small time period, we will just take a pool that survives at the highest STEP stringency, and that's because we know that such a pool will show the highest expression level. So the cells can be efficiently scaled up to any production level, and of course we will also make a cell bank to support future work, such as additional material generation or clone generation in case a customer is indeed interested in this protein that we expressed. So we believe that our STEP technology is actually also a good competitor to transient production, of which the benefits is of course the speed to rapidly generate high levels of protein. And we believe that the STEP technology brings an additional advantage as it also delivers rapid high levels of protein, but the additional advantage of having cell banks available of stable pools for future work. So on the right you see some of the products listed in which we obtain varying titers with the STEP technology in 7-day batch production. So no process optimization, no fat batch, only a 7-day production to check for volumetric productivity. And also is now here indicated in this table the stringency level at which those pools were obtained. And in most cases that was at the highest STEP stringency with titers varying from 300 to 1000 mg per liter in this 7-day batch. And in case of one very large and difficult to express blood coagulation factor, we obtained 25 mg per liter at the lowest STEP stringency levels. Cells did not survive in case of this large protein at higher STEP stringencies, but this indicates also the value of adaptability in stringency in case of difficult to express proteins that already introduce enormous metabolic loads to the cells. So I will now proceed to show you some case study material, the expression of recombinant erythropoietin, EPO. So as I just explained, the STEP technology looks promising in sense of having an efficient process with high yields, but we also performed several case studies to also better address topics like quality of the product, scalability and stability of expression. And in this particular case, we chose the EPO protein for this case study since it is a well-known protein. It is highly glycosylated, so it has 3 N-linked glycosylation sites and 1 O-linked glycosylation site, and approximately 40% of the mass of EPO is due to glycosylation, and especially proper N-glycosylation has been shown to be needed for the biological activity of EPO. So therefore this protein is a good candidate to show if our cells are also producing EPO proteins of good quality. So we generated 23 CHO-STEP pools, and on the left you see the maximum volumetric productivity that was obtained in batch cultures in shake flasks. So here each dot represents 1 cell pool, showing the maximum volumetric productivity that was obtained by that cell pool during a batch culture. So the highest producer pool produced over 300 mg per liter, and the average production level of all the cell pools was above 100 mg per liter. So to get already a first impression of protein glycosylation, from those EPO expressing cell pools we subjected supernatants to isoelectric focusing, IEF, by which the isoforms of the proteins which differ in their charge are exposed. So you see on the right some examples of supernatants and also EPREX, which is one of the actual commercial products of EPO. EPREX is composed of 1 type of protein, EPO, but it differs in its composition of glycoforms. And as you can see, EPREX contains predominantly 4 isoforms that differ in their glycosylation, with high sialylated glycoforms ensuring the biological activity of this product. It was thus important for us to assess whether our CHO-STEP pools are also able to produce those high sialylated glycoforms. And on the left 2 lanes you see 2 examples of supernatants from our CHO-STEP pools that were positive for the presence of isoforms equal to those in EPREX, and those are annotated by green dots in the graph on the left. In the right lane you see an example of supernatant that was not positive for high sialylated glycoforms, and those are annotated by black dots in the graph. So importantly, more than 70% of CHO-STEP pools was positive for highly sialylated EPO. And also important, there appears to be no correlation of expression levels and presence or absence of proper isoforms. And also important, proper isoforms of EPO are present within the highest producing pools. So we selected 2 pools based on yield and isoforms to study further. So these graphs depicted here show the growth profiles and expression levels of these 2 selected pools in shake flasks in the non-optimized 7-day batch culture. And maximum viable cell densities vary between 8 and 10 million cells per ml, and expression of EPO is above 200 and 300 mg per liter respectively. So next I'm showing a representative example of CHO-STEP performance in a batch bioreactor. This is a 2-liter bioreactor, and cells were cultured in the absence of antibiotic selection, and the run was stopped after observing a drop in viability. Maximum viable cell densities now reached 12 million cells per ml, and the batch ran for 8 days, and yield was approximately 200 mg per liter. So this is actually showing very similar behavior as we observed in the shake flasks. This is an example of CHO-STEP performance in fed batch bioreactors. Again, a 2-liter bioreactor was used, and the culture was in the absence of antibiotic selection. A generic, non-optimized feeding was applied, existing of a 5% volume feed applied at 5 time points. And maximum viable cell densities reached maximally 8 million cells per ml, and good viability was prolonged. So this fed batch bioreactor ran for 14 days. Production levels now were above 600 mg per liter. So these results show that the CHO-STEP cells are efficiently and successfully scalable. And in general, we observe yields in batch bioreactors that resemble to yields in shake flasks, and in the non-optimized fed batch we see 2 to 4 times increases in yield. Also depicted in the graph to the right is that, for comparison, a Roche patent on an originator production cell line describes titers of 50 mg per liter in a repeated fed batch, exemplifying actually the yield that we are obtaining with the CHO step technology. So early on we saw already that the IEF indicated that our EPO consists of correct glycoforms, but to address glycosylation more properly, we purified EPO from 2 bioreactors of 2 different CHO step pools, and we analyzed the N-glycans with mass spectrometry. And we also performed to an in vitro activity assay to address biological activity. So here on the top right corner, you see the mass spectra that we obtained for the N-linked glycans of our 2 EPO samples. And the 2 samples contain typical glycan structures as reported for commercial EPO products like EPREX. The 2 samples contain B3 and tetra and ternary fucosylated glycan structures with high levels of cellulation. And interestingly, our 2 EPO samples actually show identical glycan structures as reported in a Nature publication for the recombinant EPO reference protein, shown here on the bottom right. And this is indicating correct glycosylation by our CHO step cells. And interestingly is also that the glycosylation obtained on EPO by the 2 different CHO step pools is actually very similar. So those 2 EPO samples were also tested for biological activity with a cell-based in vitro assay. And in that assay, UT7 cells are incubated with the recombinant EPO samples, which then react to EPO by a linear increase in proliferation. And absorbance values after a colorimetric assay are then compared to those obtained for the recombinant EPO reference protein. And by that, relative potency can be determined. Our 2-step EPO samples showed a relative potency of 122 and 125%, which is within the required range of recombinant EPO as described in the pharmacopoeia. As an internal control, EPREX was also tested and showed a relative potency of 120%. So finally, we also wanted to address in this showcase the stability of expression. We therefore subjected the step EPO cells to a stability study, and the pools were cultured in the absence of antibiotic selection in shake flasks, and they were subjected to batch culture in duplicate at generation 20, 40, and 60. And you see that pool 1 shows stability from generation 20 to generation 60, both volumetrically and specifically, and pool 2 shows some decrease of volumetric expression at generation 60, but this is mostly leveled out by cell numbers, since specific productivity is much less reduced. So I would now like to follow with some of our key conclusions, and after that there will be time for a questions and answers section. So our conclusion is, with the STEP technology, we developed a highly stringent yet adaptable expression platform which delivers high expression CHO producers that show sustained stability of expression and are effectively scalable. The cells are directly screened in shake flask format, giving performance prediction for bioreactor productions. The STEP technology delivers a very efficient, rapid, and therefore cost-effective process because only few cells need to be screened, and the cells are directly screened in suspension formats. So a high-quality, biologically active product with correct glycosylation is generated by the CHO step cells, and because of the rapid pool generation, we also believe that the STEP technology offers an attractive alternative to transient productions with the additional benefit of having cell banks available, supporting potential future work as material generation and clone generation. We thus conclude that important hallmarks in cell line development as yield, labor intensity, speed, stability, product quality and scalability are successfully addressed by the STEP technology. So now, at the end of this presentation, there is some time available for the questions and answers section. So I'm going to check if we already received some questions. So one of the questions is whether Ceazin is allowed as selection antibiotic by authorities for its use in pharmaceutical products for human use. That's a good question, because typically actually antibiotic selection would be preferably not available or not present in any pharmaceutical production process. It is, however, allowed to be processed in any upscaling events, but from a quality perspective and a safety perspective from the authorities, it would be rather be prevented. So that's actually also why we performed the stability study. So that stability study for 60 generation actually allows very flexible upscaling potential, and within those 60 generations we still see, in the absence of Ceazin, that there's still good sustained expression. So let me see. Another question that we received is, why did you apply a non-optimized fed batch? Don't you have an optimized fed batch protocol available? That's also an interesting question, because indeed that's correct. We applied a non-optimized fed batch. But our aim with this showcase is, in this specific example, the EPO showcase, is to show what our technology delivers in CHO cells without performing any process optimization and just using basal commercially available medium. So for that reason, we also applied a very generic non-optimized fed batch process. So it shows, as everyone will know, that the sustained viability also with STEP results in increased yields, but it also shows that there's much to gain in case we will perform process optimization. And that's actually something that our USP team at Batavia Biosciences standardly is performing. So in case a customer is interested, then we of course will also deliver such process optimization, and that will allow us to even increase yields further. Another question that we received is if we can explain how 3 stringency levels are typically applied in a standard program, and is this efficient, and why not choose 1 stringency? Well, that's actually one of the assets of the technology, that we are not rigid in our stringency, so that we can get the maximum out of our cells. We still think that it's a very efficient process, because we typically start just by cloning the gene of interest in 3-step plasmids. So the 3 plasmids have 3 levels of stringency, and then we start with pooled generation. And because of this technology, we only need a small number of pools to identify a good performer. It's still an efficient process. So the pools that survive the selection pressure will be subjected in a batch production, and in every occasion that we experienced thus far, the highest stringency surviving pools delivers the highest production. So this helps in programs in which customers also rapidly want material generated, because we then directly use those highest stringency pools for production. And in case we proceed with clone generation, then we will of course choose the most suited pool to proceed into the clone generation program. Another question that we received is, could you elaborate on the mechanism behind the expression enhancing elements? Yes, so indeed there are 2 types of expression enhancing elements flanking the expression cassette of the step factors, and those different types of elements are implemented, and they were novel identified by employing a large screen of genetic elements using high stringency step factors. And actually those elements were identified by their expression enhancing functionality in a kind of a black and white screen, because in the absence of those elements, cells are actually not able to survive the stringent selection conditions employed by the STEP technology. So the genetic elements are important components of the factors because they enhance expression and actually also facilitate the use of the stringent selection system. Another question is, could the STEP technology be used in other cell lines? Yes, the STEP technology is an expression platform that can be utilized in multiple cell lines. The flexibility of the selection system by having an adaptable stringent selection system available makes it suitable to use in all kinds of mammalian cells. So the 3 stringency factors that are specifically designed to function in CHO cells, as I presented here, those are readily available also for other cell types. But in addition, we can actually apply the system such to have much more selection stringencies available in case needed. So from our experience, we know that different cell types function best at different stringency levels, and a too rigid stringent selection system will not be suitable for many cell types. This is not the case with our adaptable system, and we applied it to multiple cell types. I received a question whether we have done case studies on maps. If yes, can you summarize results? Yes, we did do case studies on antibodies, but actually those are quite limited. So we really focused on glycoproteins thus far, also on complex recombinant proteins, as we see that our STEP technology is really delivering additional increases and additional improvements in cell line generation processes that are available thus far. The antibody results that we obtained until now show relatively moderate results, so up to 400 mg per liter that we obtained with some modified antibodies, so IgG1 and IgG2 combinations of light chain and heavy chain. This is certainly a field that we are also further developing, and will also present some case study material there. How can you select a lead clone after 6 weeks without compromising the selection results is another question that we received. If I understand this question correctly, yes, with the STEP technology, we somewhat deviate from more general cell line generation processes. So in general cell line generation processes, there is an enormous heterogeneity, and expression levels can differ 2 orders of magnitude, so that results in large screening efforts needed to identify lead clones. And this therefore takes a lot of time and labor. But with the STEP technology, due to the selection system, only high expressors surviving pools are surviving. So the pools are already of very good quality, and they are low in heterogeneity. And therefore, only a small number of clones need to be screened to identify good candidate cell lines. So we therefore don't apply typical screening procedures, but directly subject a limited number of clones into a batch culture production to identify growth and production performance. And yeah, because we don't need multiple rounds of screening, we can identify those candidate clones in a very limited time frame. We actually did do a number of test runs in which we performed pre-screening in small culture formats to identify highest producers, but the results that we obtained showed that those clones were not outperforming a randomly selected small amount of clones. And these results supported the low heterogeneity composition of our CHO STEP pools and also the workflow that we apply.
We'd like to thank everyone for the questions that they sent in. It was very interesting. Thank you very much. There were some other questions that came in, but they are either too specific for this webinar or require extra information, which we will be sending to you after this webinar. Thank you very much.
Okay, thank you so much, and yeah, hopefully to another time. Okay, bye.
So that just leaves me to thank Femke for what was a great presentation and to thank Batavia Biosciences for sponsoring the session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. Once this webinar ends, today's survey will appear in its place, and we would appreciate it if you answer the questions in there. And we look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group Business Review Webinars. Thank you once again, and I hope you all have a nice day.